In the beginning, the signaling protein or ligand binds with the G protein receptor. After the ligand attaches, the inactive protein with GDP binds to the receptor. A phosphate from the cell membrane attaches. This makes the GDP GTP. This makes the protein active. Now the active protein with GTP goes to the inactive enzyme to change its shape and make it active. Now the phosphate goes from the GTP, making it GDP, to a second inactive enzyme to make it active as well. Now the GDP returns back to its normal position. Now the phosphorylation cascade occurs. The phosphate goes to activate cellular response.
The tyrosine kinase receptor system begins when two signal molecules, or ligands, attach to the ligand binding site on the tyrosine kinase receptor. This receptor is embedded in the cell membrane. Next, the two parts of the tyrosine kinase receptor come together and form a dimer. Once the dimer is formed, ATP transforms into ADP. During this process, 6 phosphate molecules are given off and attached to the tyrosine receptor. Next, 2 inactive relay proteins connect to the phosphates on the tyrosine kinase receptor. At this point, the inactive proteins become active and begin to change form. One receptor activates another and the phosphorylation cascade begins. Eventually, a cellular response results.
Now the phosphorylation cascade begins. Phosphates now go to other inactive enzymes to make them active. This then activates the cellular response, which releases calcium ions through the endoplasmic reticulum. The ligands now leave the receptors. The human nervous system is composed of billions of neurons. However, only about 100,000 to 200,000 neurons located in the hypothalamus are capable of producing hypocretin. A hypocretin, also called orexin, is used as the ligand that is used to stimulate the orexin receptors on selected neurons. The orexin receptors are G protein coupled receptors, and there are two known types, OX1 and OX2. In normal function, the orexin is released when a person is awake and ensures a wakeful state while suppressing REM sleep. The orexin binds to the G protein receptor, which induces a conformational change in the receptor. This causes a phosphate to be appended to the GDP on the G protein in the interior of the cell, thus rendering it GTP. The G protein then travels to an enzyme within the cell and binds to its active site. The enzyme then releases calcium ions from intracellular storage after hydrolyzing the phosphate on the GTP, converting it back to GDP. This in turn induces a wakeful state in the neuron. However, in the case of narcolepsy, the neurons that produce orexin die off, so it is never produced. This results in a lack of control and regulation of wakefulness and resistance to REM sleep, and results in randomly induced sleep attacks,